I guess we can get started. So today we are going to talk about ecological risk assessments. But before we talk about risk assessments, I want to talk more about the partitioning coefficients between different phases. So first, I want to talk more about the soil and the partitioning coefficients between soil and water. And then I want to talk about the partitioning between the environments and the biota or the organisms. And then before they go to the risk assessments, I want to talk a little bit about the history or the background. And then we are going to talk about risk assessment and the characterization of the risk. So first, I want to talk more about soil. So soil is a material that is composed of 5 ingredients, including minerals, soil organic matter, gas, water, and living organisms. But here I put a star for the organism because, like, based on different literatures, some literature says organism is part of the soil, but some literature doesn't include that. So I put a star here. And for the soil minerals, they can be divided into 3 size classes: clay, silt, and sand. And the percentage of each particles in the size classes is called the soil texture. And on the right side, we can see this is a 3-dimensional classification system. And we have the percent of sand, percent of clay, and percent of silt. So based on the different composition of each component, then we can describe the texture of the soil. And the most common mineral in soil is quartz. And for the soil organic matter, it is the plants, animal, and microbial residues in various states of decomposition. So basically, the organic matters in the soil, like they are the degradation products of the other organisms. And this picture shows an example of the soil organic matter, and this is humin. And we can see from its structure, it is very complex and it is very large because we know, like, the degradation products of the plants or some other stuff, like the molecules are relatively large, and we have many oxygens attached here because they are oxidized. And so anyway, so for the soil, it can be classified into different layers. So during its formation, the soil is arranged in different layers, and each of these layers is called a soil horizon. And when these layers are arranged sequentially on one above the other, and the form is called soil profile. So basically, on the right side, just show a common example. So we can see we have different layers of the soil, and in each layer we have its names, but we also have different composites. For example, for the first layer, we mainly we will have humans, and for the this layer we have like minerals with humans, and then we have like minerals and organic matter and some other stuff. So in this case, I have a question like, if you are an organic pollutant, which layer would you like to stay in this soil? Do you guys have any ideas based on the composites of the soil? Anyone wanna? Yes.
No. Sorry. Where would it naturally occur, or where would you like to stay?
Yeah, like which layer would you prefer if you are an organic pollutant? No idea.
Yeah.
So basically, we know that if you are an organic matter, you prefer to stay in the organic phase. So you are looking for the organic phases. So
What the top then?
yes, so if we look at different layers, we can see that humans, it is organic, and we have humans and organic matters. So basically, we want to stay in the first three layers because they contain organic matter. So in general, in the environment, and the organic pollutants, they are usually found in the surface layers because they contain organic matters. But generally, this is bad because we know that the animals or the plants, they usually have some interactions with the surface soil. And if the organic pollutant is accumulate or stays in this surface layer, then they will be exposed to the organic pollutant. And then what's the exact interaction between organic matter and the organic pollutants? Here we have generally we have two processes. And the first one is absorption and another one is adsorption. And the difference is that for absorption, it refers to the process that the chemical gets into another phase. So they just get into the body of the other phase. But for adsorption, it refers to the attachments of the chemical on the surface of another phase. And for the organic pollutant, it can be absorbed or adsorbed by the soil, but usually adsorption is usually dominant. So for organic pollutants, it contains both processes. And here shows an example, like we have this DDT molecule. So this is DDT, and we can see that some of the DDT molecules is on the surface of the organic matter, and some of the DDT just get into the body of the organic matter. And in this case, we can see that we have both processes. And this is important when we are extracting the pollutant from the soil, because we can see that for the surface DDT, they are very easy to be removed or very easy to be extracted. But for the DDT that is inside the organic matter, it is relatively harder to extract. So when we are doing the extraction of the organic pollutant from the soil, we may have some errors caused by these processes. They may not completely extract all the organic pollutants from the soil organic matter. Yes.
Yesterday you put the samples in the microwave.
Yes.
Is that mainly for the ones that are inside the organic matter?
Yes. So for that processes, we want to extract all of the DDT from the soil, no matter it is inside or on the surface. Yes.
I'm asking the purpose of the microwave.
Is to heat. As far as I know, the purpose of the microwave is to heat up the organic matter, so that the movement of the molecule will be faster, so they can get out faster. You have questions? No? Any more questions? No? Okay. So then we can use a partitioning coefficient to describe the distribution or partitioning between soil and water. So we have soil water partitioning coefficient, and this is noted as KD. This is measures the amount of chemical substance absorbed onto soil per amount of water, and which can be expressed as this one. So basically, KD is expressed as a concentration of the pollutants in the soil and the concentration of the pollutants in the water. And the water usually refers to the soil pure water. But note that this describes the equilibrium processes. And here I also want to say that the KD vary greatly among soils, because we have different soil that may have different amount of organic matters in the soil. So this value varies side by side. And for this one, do you guys have any questions? Then I want to also talk about bioaccumulation. So suppose we have DDT in the soil, and And we know that some of the worms, like the earthworm, they will eat the soil. So they can also eat the DDT. But the DDT is very persistent and it is also hard to be excreted. So the DDT will accumulate in the body of the earthworm. And we have some other animals that will eat the earthworm, like the moles or the birds. And when these animals eat these worms, they will also get the DDT. And because the DDT cannot be excreted or removed by these animals, so the DDT concentration will accumulate. So basically, if the DDT concentration is very low, is like here, and then the DDT concentration will increase in the worm, and then it will also have a large increase in the birds or in the moles. So in this case, we can see when the DDT concentration is low, then there is no effect and it is okay. But when the concentration becomes very large, then it will have some negative effect to the animals, which is bad. So basically, bioaccumulation is very bad for the animals that's at higher level of the food chain. And so bioaccumulation is a process of accumulation of chemicals in an organism that takes place if the rate of intake exceeds the rate of expression. And bioaccumulation can be quantified by bioaccumulation factor. So it is defined as the concentration of the pollutants in the organism. So CO refers to the concentration in the organism and divided by the concentration in the environment. At equilibrium. And here the organism and environment, they can be like different components. Like for example, we have calculated the bioaccumulation factor, which equals to the concentration of DDT in the earthworm divided by the concentration of DDT in the soil. And so for this one, do you guys have any questions? No? Now, here's a summary of the partitioning coefficient. So do you guys recall what is P0? No, it is the vapor pressure of the pollutant. And what is SW? We can say this is about water. So basically, it is the water solubility of the pollutant. And for the KH, we can say this is about water and air. So this is a partitioning coefficient between air and water. And for KOW, it is a partitioning coefficient between water and octanol. And we also have KD as introduced in today, like this is a partitioning coefficient between soil and water. And we also have the bioaccumulation factor, which is a partitioning between environments and organism. And we also have one that is KS that I didn't discuss. So this is a partitioning coefficient between water and sediment. So this is similar to soil because the sediments may also have different components. So the partitioning coefficients will change case by case. And here we have like the question we had last time, but for the last time we have some problems. So let's just look at this this time. So suppose we have this DDT and we know some of its parameters, like we know its solubility and we know its log KOW and we know its vapor pressure. And last time we realized that we cannot do some comparisons because we cannot connect the air phase to the water phase or the pure phase. So this time I found that the Henry's law constant of this PP prime DDT equals to this value. So we have the Henry's law constant and we have the vapor pressure, we have the log KOW, and we have the solubility. So in this case, where do we expect the DDT has the high concentration in the environment? So do you? We have already discussed this last time. So in general, we can see that no matter what we are interested, let's just say for the air phase, we know the concentration, let's just say it is at saturation. We have this concentration. But for the Henry's law constant, we have KH equals to air divided by water. So, and this value is very small. In other words, the concentration of DDT in the water is much larger than the concentration of DDT in the air based on the Henry's law constant. And here we also know that the log KOW is this value. So we also know that the concentration of the DDT in the organic phase or in the nocturnal phase, it is also greatly larger than the concentration of DDT in the water. So in this case, we can see that CO is much larger than CW, and CW is much larger than CO. So we can see that DDT is more likely to be stay in the organic phase, or like in the organisms, or in the organic matters in the soil, than in the water, and then in the air at equilibrium. And so for this one, do you guys have any questions? No. Then before we go to the risk assessments, I would like to briefly give some background about EPA. And so EPA refers to the Environmental Protection Agency and in the United States. And as mentioned for like last week, like we have SIP or before Spring Festival, we have like different environmental pollution events, like because of a DDT, and we also have some other events like the fire in the Cuyahoga River, and because of this very bad environmental pollution events, DDT was EPA was established in the winter of 1970. And the pictures shown here are the pictures when the Cuyahoga River got fire. We can see that like there was a boat on this river, and then this boat is on fire, and then the oil was spilled in the river, and we can see the surface was coated or covered by the oil, and similar to this picture. So this is very bad. And the EPA was established and to do some monitoring to make sure we have good environments. And there are some different programs listed here, like the Clean Air Act Compliance Monitoring Program or Clean Water Act Compliance Monitoring Program, and blah, blah, and some others. And here I want to emphasize the third one, which is Superfund Compliance Monitoring Program, and this is also called CERCLA. And the full name of CERCLA is Comprehensive Environmental Response, Compensation, and Liability Act. And the details can be found at EPA website. So I just have a link here. So we can just go to this website if we are interested in this topic. And so this program established prohibitions and requirements concerning closed and abandoned hazard waste sites. And this provides for liability of person responsible for release of hazardous waste at these sites. And it also established a trust fund and to provide for cleanup when no responsible parties could be identified. So basically it has a fund. So it is called, so this is also called Superfund. And so, like if we are going outside, we may see some of the information boards like this. And we can say this is a Superfund clean up site, and it has some of the informations. So if we are go to this site, it is not good to get into this area because this area is considered as contaminated. So far, do you guys have any questions? No? Now here is a question. How do we determine if the site needs remediation, or how do we determine if the site is contaminated? And so this process is called ecological risk assessment. And the full definition by EPA is like this is a process for evaluating how likelihood it is that the environment might be impacted as a result of exposure to one or more environmental stressors, like such as a chemical land use, disease, and invasive species. So in general, we have a stressor, and this stressor will be released in the environment. So it will have exposure to other stuffs. And then for this stressor, like for this chemical, it will also have some effect to the animals or to the plants or to some other environments. And then we want to combine the information of exposure and effect. Then we can estimate the risk that this stressor can have. So for ecological risk assessment, EPA also have a website introducing these processes. So if you guys are interested, you can look at this website. And here shows the framework of the ecological risk assessment. So basically it has 3 phases. The first phase is problem formation. In the problem formation phase, the risk assessors gathers information to determine which plants or animals are or might be risk in need of protection. For example, we may be interested in the woodcock or the bird. And then we have another phase, which is analysis. And during analysis phase, we are doing 2 things. We want to characterize the exposure, like to estimate or to monitor the concentration of the pollutants in the environment. And we also want to gather information or do some experiments to get the characterization of the ecological effects of this chemical, like how toxic it is. And finally, in the third phase, we do risk characterization. So we just combine the information of exposure and the information of effects to describe and quantify the risk. But here we can also see that after the risk assessment, we want to do some communications like with the landholders or the government, and then we can have a final plan of risk management. But here we can see that this process is not a one-way process. Like we may do some back and forth to finish it. Like for example, during some process, we may find that we need more information, or maybe other species is at more larger risk. So we may go back to the problem formation phase to define another receptor. And here we have phase 2, and for the phase 3. In phase 3, we have risk characterization. It includes 2 major components. The first one is risk estimation, and another one is risk description. So for this one, do you guys have any questions? No? One of the simple methods to quantify the risk is hazard quotient. And this one, we have been do some calculations about this in the Monte Carlo simulation lab. So hazard quotient HQ is a measure of risk and is defined as the ratio of the exposure, and this exposure could be predicted exposure. And a TRV, which is toxicity reference value for the particular type of wildlife being assessed. So the definition of HQ is the concentration of the pollutants or the chemical in the environment, or the exposure concentration divided by TRV, and the toxicity reference value. And for the screening level of ecological risk assessments, a TRV can be obtained by some of the toxicity studies. And for EPA, it also provides some regulatory documents like summarized for us, so we can just simply find this value. And if we go to the EPA website, we have this interior ecological soil screening level document. So this is solely for the soil. And because my research is about soil, so let's just say, which chemical are you guys interested in? No, cadmium. Okay, so let's take a look at cadmium. So we can find this file. So this is a screen level for cadmium. And so this file is very long. It has 90 pages, but most of them are referenced. So the important parts are only like about 20 pages. So if they go to this file, we can see something like this. So this is cadmium screening level. This is the screening level for the cadmium, and for different components, we can see that for plants, for soil invertebrates, and for some wildlife, it has different values. And so we can use this value as the toxicity reference value in the ecological risk assessment. And if we go, we can see that like they have different reference. So basically, they just summarize the previous studies and then give a reasonable toxicity reference value from the literature. So for the hazard quotient, if it is above 1, it indicates that exposure exceeds the no effect level and the wildlife may be at risk of adverse effect. So this is bad. And then we can see some of the examples, and because we have already done the calculations in the Monte Carlo simulation lab, so I think this will be more straightforward for us. So we know that the HQ is calculated using a reference mean BAF or as MLP as determined by US EPA and is based solely on the contaminants. So contaminant concentration in the soil, and the HQ can be calculated in this equation. So in this case, we still have the exposure concentration, but this will be replaced by this formula. And the meaning of each parameter are on the right side. So FIR refers to the food injection rate, and PS is the soil ingestion proportion of the diet. And MLP, it is a multiplier to estimate the concentration of the contaminant in the biota based on the soil concentration. And so basically, this is kind of like the BAF. And the TRV is the toxicity reference value. And this value can be found and also on the on this file. I'm not sure if it has the heavy metals data. Yes. So this is for cadmium, and we can also find like the TRV for cadmium, and we have the FIR and the PS and some other values. Oh, so I think I just made a mistake. So for this one, oops. So we talk about this value. So this is a required concentration in the soil. It is not the TRV. Yeah. And for this one, it provides the TRV and some other parameters that will be used to calculate HQ.
The CS was not here on your slides. The CS is the concentration in soil.
Yes. So, for example, in this case, we are interested in the DDT, and the information is summarized in this table. So let's just ignore Belgium. So we have different values for this parameter. So now we have soil sample from a site, and the DDT concentration of the soil sample is this one. So we know the soil concentration is like this. And the question is, what is HQ of wood cook? And do we need further remediation? So to answer this question, we can calculate HQ. And in this case, FIR, PS, MLP, it can be found in this table. And also the TRV can also be found in this table. And the CS is also known here. So basically, we just plug in the values. If we plug in all the values, we can get this value. And we can see that this value is greater than 1, which means that this site needs further remediation, or this site is contaminated. And this is straightforward. Now let's look at another question, another example. So we have the same setup, but here now we have soil sample from a site, and the DDT concentration of the soil samples are here. So because the soil concentration may different at different places, so the soil concentration has some uncertainties, and this is a standard deviation of the soil concentration. And then we have the same question. What is the HQ of wood cook, and do we need further remediation? So in this case, again, we want to calculate the HQ, but here we also have some uncertainties. So we are also calculating the uncertainties of, in this case, standard deviation of HQ. And we are familiar with the error propagation equations. So we can calculate the mean of HQ, which is the same as we have already calculated. And for the errors or uncertainty of HQ, we can use the triangle theory to get this result. So as a summary, HQ equals to this value. And in this case, we can see that because the HQ has some uncertainties, so it is a little bit harder to say if this value is greater than 1 or smaller than 1. And sometimes we can use t-test to do this if we have the sample size of the soil. But here I want to show another approach that we can calculate the probability of this comparison. So in this case, we are interested in the probability that HQ is greater than 1. So we want to know this probability. And for HQ, it can be expressed as the mean and the standard deviation. And we can have this empirical distribution of HQ, and this is 1. And we can say the mean is around 1.84. And in this case, we can see that the probability that HQ is greater than 1 is equals to 0.93. It's similar to this. So basically, we can see that the probability that this site is considered as contaminated is above 90%. So basically, we may say that it needs remediation because the probability is relatively large. And so for this one, do you guys have any questions?
Did you get the probability by dividing the area of the red over the whole?
Yes. So the whole area is 1, and the red area can be calculated. Yes. And then they can also have some further uncertainties. as mentioned in that lab, we know that the toxicity reference value may have some errors. Let's just say it has 20% of the error. And again, we want to answer the same question. So again, we can just plug in the values. We can get the average HQ, which is this one. And for the uncertainty of HQ, we can use the error propagation equation. We can calculate the relative error of each component. And then we can get the absolute error of HQ. And finally, we can get this result. We can see that compared with the HQ without with TRV that has no uncertainty, we can see that the standard deviation increased a little bit. So if we are doing the same thing, we can see that the probability of HQ, which is greater than 1, the probability becomes 0.89 or 0.90, something like this. We can see that the probability decreased. So in other words, when we have more uncertainties in this equation, the probability of HQ will increase. Or in other words, it is harder for us to determine if it is greater than 1 or smaller than 1. And again, we may also have some uncertainties for the PS, like the food ingestion rate. So in this case, we can also calculate or get some HQ with some uncertainties. And then we can also get a probability that HQ is greater than 1. And we can see that as the more uncertainty involved, then the probability of HQ that is greater than 1 will decrease. So in practice, when we have this probability, we may just compare this with previous studies or compare with the regulation, and to finally to manage the risk. And so for these examples, do you guys have any questions? No? Okay. So I think this is the end of the class. And because we have some time, let's just go through this part together. And so the 4 components of soil includes, do you guys remember which 4?
Air, water.
We have air, we have water, and we have, yes, soil, organic matter, and yes, yes, minerals. And we may also have organisms. And soil water partitioning coefficient is defined as yes. So the concentration in the soil and the concentration in the water at the equilibrium. So bioaccumulation can be quantified by. So for this one, I didn't talk about in the lecture. So it can be quantified by log KOW. So basically, the bioaccumulation factor is like kind of positively proportional to KOW, and the bioaccumulation factor equals to the concentration in the organism divided by the concentration in the environment. And here, circular refers to comprehensive environmental. That is reliability, clean. Yeah, I also forgot. Okay, never mind.
Comprehensive environmental response, compensation, and liability.
Yes.
Or super.
Yeah.
Yes. Super fun. And It's usually called superfund, yes. So the three phases of ecological risk assessment include so the first one is problem formulation, and the second one is analysis, and the last one is risk characterization. And ecological risk can be described by Hannes quotient, and it is defined as, you guys remember? Yes, it's defined as yes, it's defined as the concentration of exposure divided by toxicity reference value. So this is the end of the class. And finally, I want to say that the assignment 6A and 6B is due, I think, 2 weeks from now. And assignment 6A is also under preparation by Maria. And I guess they cannot start 6A until next Wednesday because we will have results. But you guys may start doing 6B. No. Okay. So that's all for it today.